Hi everyone, in this video we're gonna see preservation and maintenance of culture, which includes lyophilization, defreezing, and also type culture collection. First, the overview of microbial culture preservation is the process of maintaining the integrity and functionality of cells over time. It's essential for laboratories that need to keep stock cultures of microorganisms for various purposes, including research, education, and industrial use. The main goal is to keep the organism alive and uncontaminated while preventing any genetic changes or mutations. The methods for culture preservation includes long-term and short-term. Short-term maintenance, which means days to months. These methods are used for keeping cultures viable for shorter periods. And periodic transfer, it's the first process. This involves regularly subculturing the microorganism onto fresh media. Second, refrigeration. Cultures can be stored at 0 to 4 degrees Celsius on agar lands or plates. Bacteria can be preserved for weeks while fungi can last for months. Third, paraffin method. In this technique, agar lands are covered with sterile liquid paraffin to prevent dehydration. This can preserve cultures for several years. And fourth is tap cultures. Cultures are stored in solidified agar vials, which can keep them viable for a week or 2 weeks. And then long term preservation, which means months to years. These methods are for storing cultures for extended periods from months to many years. First, glycerol stocks. Pure cultures are suspended in sterile glycerol and frozen at minus 20 degrees Celsius. It's recommended to create multiple stocks and avoid repeated freeze thaw cycles to maintain viability. Second, cryopreservation. This is the process of freezing cultures at very low temperatures, typically minus 70 degrees Celsius or colder, using cryoprotectants like glycerol to prevent cell damage. Third, lyophilization. This method removes water from the culture under a vacuum to create a dry, stable product that can be stored for long periods. Let's detailedly look at lyophilization and defreezing. Lyophilization, which is also known as cryodesiccation, is a low temperature dehydration process. It works by freezing a product and then lowering the pressure, which allows the ice to turn directly into vapor, which is called sublimation. This process was notable used to create freeze-dried meals for astronauts and military personnel. The stages of lyophilization includes pre-treatment, freezing, primary drying, and secondary drying. Let's see pre-treatment. This is any preparation of the product before freezing, such as concentrating it or adding components to increase stability. Second, freezing. The material is cooled below its triple point to ensure that sublimation, not melting, will occur in later stages. Creating larger ice crystals at this stage helps make the process more efficient. Third, primary drying. The pressure is lowered and heat is applied. About 95% of the water is removed through sublimation. And fourth, secondary drying. This stage removes any remaining water molecules. The advantages and disadvantages of lyophilization is advantages. The process causes less damage to the substance compared to other dehydration methods, and flavors, smells, nutritional content generally remain unchanged. The resulting product has microscopic spores that allow it to be rehydrated quickly and easily, which is especially important for pharmaceuticals. Disadvantages: it is about 5 times more expensive than conventional drying and may not kill all spoilage organisms. There is also a risk of contamination from silicone oil leaks from the freeze dryer's heating system. And let's see deep freezing. Deep freezing preserves microbial cultures by rapidly freezing them at temperatures from minus 50 degrees Celsius to minus 196 degrees Celsius. This halts the metabolic activities of the microorganisms, keeping them dormant but viable for future use. The principle and procedure: The core principle is that at very low temperatures, biological and enzymatic reactions stop completely to prevent cell damage from ice crystals. Cryoprotectants like glycerol or DMSO are added. These substances lower the freezing point and prevent the formation of large, damaging ice crystals. First, culture preparation. Microorganisms are grown in a broth medium until they reach a viable growth phase. Then, addition of cryoprotectant. A sterile cryoprotectant solution, example 15% glycerol, is mixed gently with the culture. Freezing. Vials of the mixture are rapidly frozen either in an ultra low freezer at minus 70 degrees Celsius or minus 80 degrees Celsius, or by immersing them in liquid nitrogen at minus 196 degrees Celsius. Storage and revival. The vials are stored undisturbed. When a culture is needed, a vial is quickly thawed in a 37 degrees Celsius water bath and transferred to a fresh growth medium for revival. The advantages: it also allows for long term preservation, which means 10 to 30 years, maintains genetical stability, has a low risk of contamination, and requires a small storage space. And disadvantages: it includes the required equipment such as deep freezers and liquid nitrogen tanks, which is very expensive. Some sensitive organism may still be damaged, and there is a risk of sample loss if the freezer breaks down. Cryoprotectants can also be toxic if not handled properly. Next, let's see type culture collection. Type culture collection is a repository or library where pure reference strains of microorganisms are preserved. These collections includes like the American Type Culture Collection, ATCC, and Microbial Type Culture Collection, MTCC, and Gene Bank. In India, serves as banks for microbes. The objectives of these collections include preserving microbial diversity for long term use, providing authenticated strains to researchers worldwide, preventing the loss of strains due to mutation, contamination, or lab accidents, supporting various industries including medicine, agriculture, and biotechnology with reliable microbial source. These collections play a vital role in research, medicine, and industry by ensuring the availability of standard and authenticated microbial strains. Thank you.